Every frog knows that many crops depend upon wild pollinators. But the pool of wild bees that visit crops is actually quite small. Only 2% of wild species are responsible for about 80% of crop visits. So therefore, it really makes a lot of sense to try to understand the factors that affect this small pool of wild bees as they provide pollination services to crops. Most farmers that grow commercial-scale flowering crops face a dilemma. On the one hand, they depend upon pollinators, mostly bees, such as the bee pictured here on a pumpkin flower, to pollinate their crops. On the other hand, they must protect their crops from insect pests, such as the striped cucumber beetle, pictured here. In monocultural systems, farmers depend upon insecticides for crop protection. However, those insecticides may also harm the pollinators they depend upon. This is especially true of systemic insecticides, such as neonicotinoids. Most wild bees are solitary. This holds true for the species that visit crops also. Of the solitary bees, most bees nest in the ground. Ground nesting bees, like other bees, consume nectar and pollen and feed it to their offspring. However, ground nesting bees also handle soil as they excavate their nests. This is especially problematic because a study that we did in 2019 clearly demonstrated that Ontario's agricultural soils have high levels of pesticides in them. My name is Susan Chan. My co-author, Nigel Rain, and I studied the population decline in a ground nesting solitary bee following exposure to a neonicotinoid-treated crop. This work was published earlier this year in Scientific Reports. We used as our model system the solitary Hori squash bee, pictured on the left, and Cucurbita crops such as pumpkin and squash that this bee is closely associated with. The Hori squash bee is a strict pollen specialist on plants in the genus Cucurbita, meaning that it must collect Cucurbita pollen to feed its offspring. Interestingly, this bee is entirely dependent upon cultivated Cucurbita in northeastern North America, where we have no wild Cucurbita. Hori squash bees obtain all their pollen supply from these crops and in turn move pollen from the staminate flower, pictured on the left, to the pistillate flower, pictured on the right, to enable the production of fruit, such as this acorn squash. Hori squash bees are ground nesters who can form large nesting aggregations. Here you can see an aggregation on a lawn at a farm in Guelph that has more than 3,000 nests in it. This aggregation is safe from exposure to pesticides and tillage. However, Hori squash bees commonly nest within Cucurbita cropping areas. Each one of the pink flags you see here marks a Hori squash bee nest. This aggregation was created the year before when this sweet corn field was in pumpkin production. The bees then emerged the next year from their nest to find themselves in a sweet corn patch. The nests of Hori squash bees look like this. They're excavated and occupied by a single mated female and her offspring, shown here in the nest cells. In our 2019 study, we found that the adult females were exposed to insecticides in Cucurbita growing systems via consumption of nectar, contacting pollen as they provisioned their nest cells, and through their contact with soil as they built and excavated their cells. By far, the most hazardous route of exposure was contact with soil. In this study, we had two objectives, so I'm going to go through the objectives one at a time and talk about them separately. So objective number one was to assess the effects on Hori squash bees of exposure to crops treated with systemic insecticides. So you'll note that we did not directly expose the bees, but we exposed the crops and then exposed the bees to the crops. We carried out our study at a site in Peterborough County, where we erected 12 net-covered hoop houses. This allowed the plants to have access to the outdoor environment, and so the growing conditions were exactly the same as the outside. We applied 4 treatments to the acorn squash plants grown in these hoop houses, with 3 hoop houses randomly assigned to each treatment. The first treatment was a control with no insecticide applied. In 3 treatments, imidacloprid was applied directly at planting to the soil using the product Admire. In 3 other treatments, we applied thiamethoxam as a FarMore seed coating, again at planting, obviously. And in 3 other hoop houses, we applied Coragen-chlorantraniliprole as a foliar spray applied at the 5-leaf stage, and we used the product Coragen. In all the hoop houses, the insecticides followed the labels and government recommendations from the Ontario Ministry of Agriculture. Inside the hoop houses, we grew 28 acorn squash plants, and these squash plants were managed in accordance with the commercial recommendations from the Ministry of Agriculture. We then introduced a founder's population of Hori squash bees in early August to the crop, 8 weeks after we had applied the insecticide treatments, and that founder's population was introduced in 2017. We then continued our experiment through to 2019. We introduced the bees by capturing 96 mated females from the nesting aggregation pictured earlier on that lawn, and only female bees that were entering a nest with a full pollen load were captured, and we picked them up gently. It was really easy, and we inserted them into an aerated microcentrifuge tube. The tubes were then placed in a cooler and transported to Peterborough, where they were kept in their fridge overnight and then introduced into those hoop houses in the early morning when the crop produced a new flush of flowers. To determine the effects of exposure to systemic insecticides on the Hori squash bee, we measured 3 variables. The first variable was the number of pollen grains left on the flowers. You'll note that we didn't measure the amount of pollen that the bees harvested directly, because that would have interfered with the bees too much. To measure the pollen grains on the flowers, we collected them after they had wilted at the end of the day. These flowers last a single day, and then a new flush of flowers is produced the next day. And we removed the anthers, scraped off the pollen, and counted the pollen in the lab. The second thing we counted, or the second variable that we measured, was the number of nests produced by the bees in each hoop house. This was easy to do. The bees were really easy to find as they made their nests, and we marked each active nest with a marker like this, and each nest was given a unique marker. And so at the end of the season, we were able to collect all the markers and count the number of active nests that there had been over the season. The last measure we used was the number of bees or offspring that were emerging from those nests. We had to do this in the year following the year that the nests were made, because these bees produce 1 generation per year. We measured the number of bees and the sex of the bees that emerged the next year in each hoop house. We found that bees in the Admire or imidacloprid treated hoop houses left about 5 times the amount of pollen unharvested on the flowers than bees in the control, meaning they collected much less pollen. The bees in the Coragen-chlorantraniliprole treatment also left significantly more pollen unharvested than the control, but there was no significant difference between the control and the thiamethoxam treatment. Not only did the bees exposed to the imidacloprid treated crop harvest less pollen, they also made 85% fewer nests than did bees in the control in both 2017 and 2018. 2017 is marked in green, 2018 is marked in gray. There was no significant difference between the other treatments and the control in both years. As a result of making fewer nests and harvesting less pollen, it is unsurprising that Hori squash bees in the Admire or imidacloprid treatments produced 89% fewer offspring than did the control. On this graph, the green bars represent the 8 mated females introduced into each hoop house in 2017. You will note that the populations declined in the Admire-treated hoop houses, whereas they increased in all the other treatments. There was no significant difference among the control, Coragen, and FarMore treatments in each year. Our second set of objectives were related to production. So we wanted to determine if the effects of exposure to systemic insecticides impacted yield. And our reasoning was that if the bees were being impacted by the systemic insecticides, then they might not be able to provide as much pollination service to the crop. So we measured this by counting the number of acorn squash that were produced in each hoop house and weighing them, and all the fruit that were less than 500 grams were discarded as unmarketable. We defined a marketable fruit, as I said before, as one weighing at least 500 grams. To obtain a marketable fruit, bees would have had to pollinate the flower, and the plant would have had to devote resources to its development and maturation. We found that in all treatments, more than 50% of the fruit were marketable, and there was no significant difference among the treatments. This means that in all treatments, bees were foraging on the flowers and moving sufficient pollen around to affect pollination. Although their reproductive activities were impacted, they were still foraging. And if a grower were to evaluate the health of the pollinator community based on these yield results, they would find no problems. Another thing you'll note on this slide is there was a significant difference between the 2017, the number of marketable fruit in 2017 and the number of marketable fruit in 2018. This is attributable to a change in the variety that we had to use, and I'd be happy to explain that to you during question period. So what are our conclusions? Well, first of all, we know that exposure to imidacloprid treated crops has a negative impact on pollen harvesting, nest establishment, and reproduction, and this results in population decline. So our conclusion is that application of imidacloprid to soil should be discontinued. We also did not observe impact on production indices in any treatment. So our conclusion is that production indices are really not a good way to evaluate pollinator health, because it's very clear that pollinator health was being impacted, yet pollination services were not. And these pollination services were not being impacted within a season. Over the long term, we would expect pollination services to decline. At this time, I'd like to thank the University of Guelph for hosting this research, and the fresh vegetable growers, the Ontario Ministry of Agriculture, Food and Rural Affairs, and the Weston Family Foundation for their financial support for our work. B. Chan, Dylan Muldoon, Lauren Moran, Leonard Zettler, Matt Gibson, Katie Fisher, and Emma Jane Wood all contributed to this project, and I'd like to thank them. Now I'd be happy to field questions if you have any.